Welcome everyone to today's webinar. My name is Dr. Maria Morgan Bathke. I am the chair of the Research Dietetics Practice Group, and I will be serving as the moderator for today's program. Today, we aim to provide some clarity around the current understanding of the role of nutrition in COVID-19, both common and emerging, that are central to the conduct and understanding of gut microbiome science. We are very pleased to offer this webinar to you as a collaboration between three groups as shown here. IFANS, or the Institute for the Advancement of Food and Nutrition Sciences, is a nonprofit scientific organization that advances food safety and nutrition science for the benefit of public health by bringing together scientists from industry, government, and academia. The Academy of Nutrition and Dietetics Research Practice Group brings together registered dietitian nutritionists with a particular interest in nutrition-related research, and any RDNs in the audience with this interest are encouraged to join for additional value and benefits of membership. I'm honored to be a member of this practice group and to serve as your moderator for this excellent program. Finally, the American Society for Nutrition's Nutritional Microbiology Research Interest section brings together investigators who explore interactions between dietary compounds and the microbiome. Our focus is on fundamental and translational research to address pathological conditions and preventative measures and to enhance human performance. This webinar is approved for 1 continuing education credit by the Commission on Dietetic Registration. Access to the Certificate of Participation will be provided to participants shortly after the webinar. Let's quickly touch on some logistics. On this slide, you see the control panel, which should be visible on your desktop. We encourage you to submit questions throughout the program by typing them into the questions box. Only the organizers will be able to see those questions. Before we begin, we wanted to get a sense of the audience by trying out a live poll. Before we show you the poll question, the instructions are on this slide. You can find the poll by either going to the web address shown or by using your phone and first texting iFans Connect 300 to the number shown. We'll give people a second to choose one of these options. So our question is, how established is the science related to nutrition in the severity of COVID-19? And we're going to give folks a second to respond here. We've got some options. Specific recommendations are available. Nutrition has proven benefits. Nutrition has likely benefits. Very little is known, or none of the above. We're starting to see some responses come in. It looks like about half of folks that have responded so far are saying C, nutrition has likely benefits. About 30% of folks are saying B, nutrition has proven benefits for COVID-19. About 25% are saying very little is known. Still seeing some folks answering here. It looks like our responses are slowing down. So it looks like about half of the audience believes C, nutrition has likely benefits. About 25% are saying B, nutrition has proven benefits. And D, 25% are saying very little is known. So just barely a majority are going with C. So thank you everyone for engaging with us on that question, and hopefully our webinar today will help us understand a little bit more on what we know. So let's get right into today's program. We're hoping that participating in this webinar, you'll be able to describe how characteristics of the gut microbiome impact COVID-19 outcomes and dietary components that can support immune responses, as well as the state of this evidence. And with that, I'm pleased to introduce our speaker, Dr. Liam O'Mahony. He is currently the professor of immunology in the departments of medicine and microbiology, APC Microbiome Ireland, the National University of Ireland in Cork, Ireland. And his research interests are focused on the molecular basis for microbe and metabolite modulation of mucosal inflammatory responses. And we'll send it over to you.
Great, thank you for the introduction, Maria, and thank you for the invitation to speak to you today about what I think is really an incredibly important topic of the role of the gut microbiota and nutrition and the interaction between the microbiota and nutrition to the risk of getting COVID-19 and also the severity of COVID-19 and perhaps even the longer term consequences into long COVID. So what I will do today is I'll start off with a very general introduction into why I think the microbiome is very important for proper immune function. I'll then talk about some of our work in allergy and asthma, and I think that is a little bit ahead, of course, of our understanding of the microbiome immune system interactions in disease. And some of the aspects that will be really important to get for when I get to talk about COVID-19. And then at the end, you will see that we have still a lot more questions than what we have answers for. And I really appreciate you putting questions in and comments in about how can we move forward perhaps with future interventions to impact the microbiome through nutrition and increase protection against the severe effects of respiratory infections like with SARS-CoV-2. So these are my disclosures. So I'm a full-time employee at University College Cork. And just to start, so with the human immune system, we all know, and you all know, that it's important for protection against infection. But it does a lot of other things. So it repairs damaged tissue. It's very important for metabolism. It regulates metabolism. And I think for the purposes of today, it's keep in mind that the immune system is actually a communications platform. And it's really important to integrate signals that come from outside the body, like from the diet and like from the microbiome, into the overall physiology of the host. It's present in all organs of the body. It's one of the unique features about the immune system. It's everywhere. It has extensive sensory capabilities, so lots of different receptors, lots of different mechanisms to see what's there and what is it doing. It then has a vast array of molecules for communication and for effector functions. And one of the, again, a unique feature about the immune system is it has the ability to learn, adapt, and remember. And of course, that's a basis of vaccination. And finally, the majority of human diseases actually do have an immune component. So I think it's incredibly important that we understand how the immune system is regulated, particularly by factors that are external to the body, like the microbiome and nutrition. So when we study and we look at different cells in the immune system, we often in the lab look at individual cell types, from the adaptive cells to the effector cells and the innate cells. But in vivo, in real life, all these cells work in concert with each other. And when you launch an immune response to a virus like SARS-CoV-2, all of the communication and coordination between these cells is really key. So in this picture, who's the conductor? So we would suggest that the microbes within your gut actually play a huge role in directing this coordination between the different aspects of the immune response. So just a few words on the microbiome. So these are the bacteria living in us and on us. There's a lot of bacteria, so at least half the cells in the human body are not human at all, they're microbial. And if you add up all the genes associated with the microbes living in you and on you, it adds up to about 2 million genes. And if you contrast that with the human genome of 20,000 genes, that means genetically speaking, we're 99% microbial. And these microbes are very metabolically active. And this is a theme that I will come back to a couple of times during my talk today. So these microbes utilize body secretions like mucins, but also particularly in the gut, they utilize dietary substrates. and they can generate literally thousands of different molecules from their utilization of these substrates. Many of these molecules are small molecules, so they can be absorbed and they can have effects at organs distant to where they're produced. So that's why, for instance, a microbe in the gut metabolizing a food source can have effects on what happens in the lungs, because the metabolites produced in the gut can be absorbed and can have direct effects on other tissues. And as an immunologist, I've always been fascinated by, how does the immune system not react aggressively to the presence of all these, foreign microbes that are living in us and on us. So, the classical view of the immune system, that it's there to react and protect against everything that comes from outside the body, is actually wrong. The immune system is very well regulated usually. There's potent tolerance mechanisms in place that prevent these overreactions. And I think by understanding those tolerance, these natural tolerance mechanisms, we can really unlock a lot of what might be going wrong in immune-mediated disorders in general, but also in infectious diseases. So as I said, there's a lot of bacteria in us and on us. In total, there's about 1,000 different species have been described to be present in the human gut, but any one of us at any one time, we don't have 1,000 species. We have maybe about 200 to, at tops, maybe 250 different species. And as I said, there's a lot of genes associated with these microbes, and they make a lot of different metabolites. But one of the questions is, why do we need so many different microbes? And actually, there could be a very good reason from a nutritional perspective, in that as humans, we lack a lot of the enzymatic machinery to break down many of the foods, particularly the plant-based foods that we eat. Whereas the microbes can produce, again, literally thousands of different enzymes that really allows them to handle, many of the different types of foods that even historically perhaps were more important than what they are today. So I'd like to show you this very simplified model for how diversity in the microbiome and diversity in food may be very important for immune function. So on the x-axis we have time, and on the y-axis I'm going to show you bacterial numbers. So let's say, today you're eating one type of food, and there's one type of bacterium in the gut that likes this food. It metabolizes the food, the bacterial increase in number. And tomorrow you eat a different type of food, and there's a different type of bacterium that likes this food, and this bacterium metabolizes the food, generates the metabolites from that. And the bacteria from yesterday that was eating the food type 1, this doesn't go back down to 0. This is still there. And then as you go on through the week, again, you're eating different types of foods, and different type of microbes will preferentially utilize the substrates that are present in those foods and thrive. So this is a model for, diversity, both from a food and also from a microbial perspective. But from the immune system, actually, this is quite challenging, because every day you're seeing different antigens, you're seeing different foreign proteins, both from the food as well as from the microbes that are increasing in numbers. But also there's a lot of danger signals that are classically associated with microbes, like LPS, for instance, or many other innate triggering substances. So the immune system is constantly looking at, the immune system isn't ignorant of these changes. It is sampling and it is sensing these changes, but it doesn't overreact usually. So I think what that means is, also with these very different immune challenges or immune stimulations, there has to be some tolerance adjuvants that come along with that. So these are adjuvants that would help to calm down the immune system and prevent it from overreacting. And what that means overall, if you've a varied diet and you have a diverse microbiome, when you have all these changes happening all the time, you're promoting an immune system that's very, very robust, because you're stimulating all the effector functions, but you're also stimulating all the regulatory functions. So the immune system can react very aggressively and very quickly when it needs to, but normally it is very, very well controlled. So this kind of diversity gives a nice redundancy into the system, and I guess that's the key word that the immune system benefits from. But what's happening in post-industrialized civilizations. So food diversity is decreasing, particularly within the plant food categories, but also the types of microbes that we have are also decreasing. And actually, some of the more recent surveys have shown in the USA and in Europe up to a 30% loss in microbial diversity. So what that might mean is you're missing key microbes to metabolize specific foods, or you're not eating a specific food substrate anymore that's allowing that microbe to thrive. And for the immune system, that means you're missing some triggers for effector functions, and you may also be missing some tolerance type of adjuvants that would allow the immune system to be well controlled. And in the allergy and asthma world, there's been many studies looking at the microbiome on the skin, in the lungs, in the gut, and consistently there's been shown to be differences in the composition and their metabolism associated with atopic dermatitis, with asthma, and with food allergy. So changes in the microbiome and in their metabolism associate with inappropriate immune reactivity at different sites in the body. And I'm just going to give you a couple of examples from our own work. So this is a study we did looking at obese asthma patients. And we didn't just look at what was different in asthma between those who had asthma and those who didn't have asthma, but even if you look at those who have severe asthma, so this is asthma that is largely uncontrolled with corticosteroids, for instance, we found that this microbe, if I can just give me a second, this microbe shown here was actually missing in those who have severe asthma. And this was Akkermansia muciniphila. So Akkermansia, normally about 5 to 10% of all the microbes in your gut are Akkermansia, and this is missing in those with severe asthma. So we took Akkermansia, we put it into mouse models, and we could show that allergic airway hyperresponsiveness is reduced in those animals that got Akkermansia. And if we look at all these inflammatory cell types in the lungs of mice, which we challenge with house dust mites, so it's a relevant trigger for inflammation that correlates with many of the features of asthma in humans. Feeding with Akkermansia reduces a lot of these features. So Akkermansia could be one microbe that you would think might be protective against aberrant inflammation in the lungs. But what about the metabolites that are being secreted by the microbes? So histamine is a metabolite that we all know very well. But histamine doesn't just come from human cells. There's many bacteria in the gut can make histamine. And we were isolated a number of different strains from humans, from the human gut that can make histamine. And some of these strains make actually huge amounts of histamine. So we went back to our asthma cohort and we looked at those who have asthma, who don't have asthma. And people with asthma have more bacteria or a greater level of bacteria that can make histamine. And the main one that seemed to come out from our screen was a Morganella morgani strain. And again, there's more of this microbe in asthma patients. And this was really an incredible microbe that from a metabolism point of view, if we put 10 grams of histidine, the amino acid, into the culture media, you've 10 grams of histamine the next day. So it's really good at bioconversion of histidine to histamine. And actually, if we divide up our asthma patients into those who are mild, moderate, or severe disease, those people with the highest levels of Morganella morgani all have severe disease. Now, not everyone with severe disease has high levels, but in this subset, you would wonder, are these symptoms, which are non-responsive to corticosteroids, are these symptoms perhaps due to, or at least influenced by, this high level of this microbe within the gut that's secreting a metabolite that is perhaps causing some of the symptoms within the lungs? So that's just one example of a microbe so I gave you one example of a microbe that might be protective for the lungs, and also here's an example of a microbe that might be detrimental. And histamine binds to a class of receptors in the human immune system called G protein coupled receptors. And there's many of these, there's about 800 different G protein coupled receptors. So what we've done in this study is we've basically been mining microbes present in the gut, in the airways, and in the skin for metabolites that could bind to human G protein coupled receptors. And I guess the takeaway from this, red basically means the presence of microbes that can make these different metabolites. And we screened over 1,300 human gut samples, 122 airway bowel samples, and over 280 skin samples. And I think what's really interesting is a lot of these metabolites are made not just in the gut, but also in the lungs and on the skin. So it suggests to me that there's really this core metabolic output from microbes that survive in close proximity to the human system. And it doesn't matter if that's in the skin or in the gut or in the lungs, that actually serve to dampen the immune response. So this is why, that's why we're focusing on some of these metabolites in our studies in COVID patients and also in others to see if there are more or less of these metabolites. So let's go away from metabolites for a moment, but let's think more holistically. So how do you develop a microbiome community in the first place? So you're born sterile, and the type of factors that influence microbiome development in early life are shown here. And the two key ones that I'll talk about are there's different dietary patterns that are really important for the maturation of an appropriate early life gut microbiome, but also there's early life exposures that are really important. So you need to get the microbes from somewhere. So I'll just spend a few minutes just to talk a bit more about those two aspects. So one of the most important, at least that we know so far, dietary influences on early life microbiome, but also the adult microbiome, are the non-digestible fibers in the diet. So these are not digested by us. They travel into the colon, and they're good substrates for microbes to ferment. And the results of this fermentation lead to many metabolites, as I've already said, but the ones that we know that are really important so far are the short chain fatty acids. So these are acetate, propionate, and butyrate. And these also bind to G protein coupled receptors in the immune system. And butyrate can also have epigenetics effects on immune cells, whereby they really support the tolerogenic or the regulatory arms of the immune system. So here you have an example for if you're eating a lot of fibers and you have the right bacteria, you're going to be generating a lot of acetate, propionate, and butyrate in the gut. That's going to have a very profound effect on the immune system. So we already knew that early in life that the consumption of fruits and veg and yogurts in the first year of life is protective against later life allergy and asthma. So in this Pasteur-Ephraim Burt cohort study from Europe, we also measured at year 1 fecal levels of butyrate, propionate, and acetate. And basically what I've divided it up here into is those kids with the lowest levels of butyrate versus those kids with the highest levels of butyrate, and then the same comparisons for propionate and the same comparisons for acetate. And on this scale here, what I'm showing you is any sensitization to any allergen by school age, so 6 years of age. And what you can see quite clearly is those kids that started life with a very high level of butyrate, much fewer had an alert or an IgE response to an aero or a food allergen by school age, and the same is true for propionate. So we did a lot of studies around this, and I'm just going to summarize that here, that diet early in life characterized by high levels of yogurt, fruits and veg seem to be really important for the generation of these short chain fatty acids by the microbiota, which leads to an improvement in the regulatory immune response early in life, and that's associated with a lower risk then of atopic sensitization, asthma, and food allergy later in life. But what Karina Venter has recently published is this dietary characteristics are not just protective in the infant, but may actually start much earlier. So what she looked at was these dietary patterns during pregnancy. And actually, if there's more fruits and veg and yogurts consumed by the pregnant moms, their offspring have a lower risk of allergy and asthma. So we would propose, and this is a study that she's discussing at the moment to hopefully start soon to test, but we would propose that this dietary effect is mediated by the microbiome, and that metabolites made by the pregnant mom's microbiome in the gut can actually travel and have effects in utero. such as the epigenetic effects that I mentioned previously. So the other, as well as diet, we also need to get microbes from somewhere. And there are a number of different ways you can get microbes when you're early in life. So of course, from the mum is a very good source. Also from other people in the home, so older siblings are very good sources of microbes, as are the wider community and family structure. So aunties, uncles, grandparents, and so on. Everybody will contribute microbes to the developing infant gut microbiome. And something that we're just realizing is actually that environmental and animal exposures are also really important for that early life microbiome assembly. So one of the studies we did recently that really brought that home for us was this was a study we did in South Africa in rural and urban kids who had atopic dermatitis or who didn't. And you can see very clearly in those with atopic dermatitis, they have higher levels of IgE, as you would expect, to food allergens. And this is true both within the rural farm environment or the urban township environment. So there's nothing surprising there. And also if you look at other antibody levels, but really the surprising factor was when we looked at a lot of the cytokines in these kids, these are the circulating immune mediators and communication chemicals, actually location had a huge effect. So the rural kids, whether they had AD or were healthy controls, had higher levels of this cytokine IL-8 compared to the urban kids. And it was the opposite when we looked at other cytokines like IL-22, an important barrier cytokine. So while there are some immune effects associated with disease like AD, a lot of the immune programming is actually associated with where these kids are living and where they're being brought up. And Notlantla had done a lot of work characterizing the living environment of these kids. And what she could show was a lot of these changes were due to farm animal exposures and pet exposures, as well as other lifestyle factors, but these were really key. So again, just to bring back this idea that exposures to diverse sources of microbes are really important to build that early life microbiome appropriately, and also to generate these metabolites that can have an influence on immune function. But what has happened over the last 2 years then? We've all lived through a period of very hard lockdowns. And here in Ireland, we had a category 5 lockdown, for a lot of the last couple of years. So these kids that are born during this time, where are they going to get their microbes from? And this immune development, therefore, may be very different in children who are not exposed to people or not exposed to diverse environments compared to those beforehand. And this may associate with a more hyperreactive immune system now as well as later in life. So this is a study that we have ongoing. It's called the CORAL study. In March 2020, we started recruiting infants born during the first lockdown in Ireland. And these now are nearly 2 years of age, and their 2-year assessments are being done right now, actually. But we've been looking at their microbiome, we've been looking at their allergy rates, we're doing a lot of immune measurements with them and dietary evaluations and so on. And I don't have all the data to share with you today, but I'll just give you some top lines for what we do know. Just from a dietary point of view, so breastfeeding rates were really way higher than what they would normally be in an Irish population during this lockdown period. So that's a good thing. But when we look at the introduction of potentially allergenic foods into these kids, and as we know, the advice has changed to that we shouldn't delay introduction of potentially allergenic foods. And in fact, it may be protective to some kids to have that early introduction. But if you look at foods like peanuts, still only 13, 14% of the kids have had an introduction of peanut protein into their diet by 6 months of age. So that's much, much lower really than where we would like it to be. But this was the same pre-pandemic as during the lockdown, so this didn't change. But if we look at the exposures of these kids, and I just give you some examples here for some of the things we could show, their exposures are completely different. So on average, 3 people will have kissed each of these infants by 6 months of age, and that includes their parents. And 1/4 of these infants, when they get to 1 year of age, have still not met another child of similar age. So if you think of microbial exposures, these kids will be very, very different. At their 1 year allergy workups, we found that 7% of these kids are sensitized to one or more foods. And if we compare that to a baseline cohort we have here in Ireland from just over 10 years ago, which was 4%, this is a very significant increase. And if we look at atopic dermatitis, it's even more dramatic. So 15% in our baseline cohort, but 25% now in the kids born during lockdown. So this does suggest that there is something going on with their microbiome and immune system, even though a large number of them were still being breastfed, and very few of them required antibiotics, at least early in life, which are two very different aspects than what we wouldn't have seen before. So the microbiome and all the immune analysis is currently ongoing. I don't have the data today to show you this, but hopefully in the not too distant future, I can share that data with you. But what about in adults with COVID? Excuse me. So the group that I'll talk to you about today is a group of hospitalized patients that we have extensively worked up. So it's just under 200 patients that were hospitalized with COVID. Our control group was 29 age and gender matched and environment matched people. And of the people with COVID-19, 42 were hospitalized but ended up having a mild or moderate type of COVID. 130 had severe COVID, so this is ICU, oxygen support COVID, and of those 41 had a fatal outcome, whereas 89 survived. Now, this is the group that really the important comparisons are made in, because within this group, the survivors and the non-survivors are well controlled for each other regarding age, other comorbidities, and so on. So any of the molecular differences that we see in this group could really be inferred to really directly impact on the severity of disease. So we looked at a whole range of different cytokines in these patients, and I guess this heat map red basically means significantly increased versus the comparators. So anyone with COVID that's hospitalized versus controlled, there's a huge number of cytokines that are different, that's well known. If you look at those with severe versus mild moderate disease, it's still a lot, but it's less than versus controlled. But if you look at those with a fatal outcome versus those with severe disease that survive, there's a smaller group of very, very interesting cytokines like TSLP that's released from damaged epithelial cells, or IL-15 that's associated with a natural killer cell response to the virus. The other thing I should mention is all these samples were taken from the first sample following hospitalization. So usually within 24 hours of these patients coming into hospital, that's the results that I'm showing you today. So very, very early differences in the immune system. And this is important because if you look at the typical inflammatory markers that we'd look at to see if someone has a disease or inflammatory disease or not, like CRP, C-reactive protein. This comes from the liver. You can see there is and this is a log scale. There's a very nice increase in patients with COVID, whether they have mild, moderate, or severe disease. This is what you'd expect, but it doesn't discriminate between those who survive or who have a fatal outcome. Whereas those cytokines I mentioned, like TSLP or IL-15, they actually do discriminate those who have a fatal outcome versus those who survive. And their levels are higher in those who have a fatal outcome at this very early time point following hospitalization. And the reason as well I'm really highlighting these two cytokines for you is in our long COVID cohort, where we started to look at some of these cytokines, these two cytokines, TSLP and IL-15, remain elevated for a long time. So this is greater than 6 months following hospitalization for COVID-19. And these patients all have long COVID. So while the typical inflammatory markers like CRP are all back down to normal levels, these really specific molecules within the immune system are still elevated. The other aspect that's really important is the gut barrier. And we measured this, and that's because even though within the gut, the microbes are really important for immune health, if they're released from the gut into the systemically, they will induce a very potent pro-inflammatory response or a sepsis-like response. So what we did is we measured bacterial DNA levels in peripheral blood. We all have a little bit of bacterial DNA in peripheral blood, but these levels really increase a lot in someone with severe COVID-19, and it's even higher in those who had a fatal outcome. But this is really a temporary effect. So if you look here of weeks following infection or weeks following hospitalization for the infection, while the levels are very, very high at the beginning, they come down. And if you look in those with long COVID, bacterial DNA levels are also back down to normal levels. So this is really an acute failure of the barrier that happens very early in the disease. But with those who survive, it comes back down to normal levels. We also looked at the metabolism of these patients in great detail. We looked at over 1,000 different metabolites. I'm obviously not going to go through everything, but I think you can appreciate from this heat map, if you look at healthy people here, these dark colors mean that they're less, and people with severe COVID, all of these metabolites are increased. So you see this huge reprogramming of host metabolism. And just the one metabolite that I want to highlight for you is tryptophan metabolism, because this is really one of the key amino acids that is metabolized both by the host as well as the microbiota. So in the host, tryptophan is metabolized on one side into serotonin, but it can also be metabolized in an alternative pathway into kynurenin and quinolinic acid, whereas the microbes in the gut metabolize tryptophan to generate these indoles. And these indoles are really important ligands for the aryl hydrocarbon receptor in immune cells. And these actually serve to dampen a lot of the inflammatory responses and improve the gut barrier through these indoles signaling in immune cells. Whereas from the host point of view, you want to have more of the serotonin, because this is a neuroprotective metabolite, and you want less of quinolinic acid, because this is actually a neurodamaging or a neurotoxic metabolite. So what happens in COVID patients? So actually those with fatal or severe disease, you have much lower levels of serotonin. So serotonin production from tryptophan seems to be turned off, whereas the kynurenin and the quinolinic pathway is being increased, so you have more of these. And from the gut microbiome, we have less indoles, particularly in those with severe and fatal disease, being made from tryptophan. So you can see this really profound dysregulation of tryptophan metabolism. And this is also long lasting. So if we look in our long COVID patients, we still see less serotonin and more quinolinic acid. And given that this is a neuroprotective and this is a neurotoxic compound, this may underlie, which is being investigated right now, a lot of the symptomology associated with fatigue and brain fog. So finally, the microbiome in these patients, what happened to those? So we were able to see, if we looked at everyone together who had COVID, that the microbiome configurations really split into two groupings that I'm calling microbiome group 1 and microbiome group 2. And if we looked at what differentiates these, and the microbiome group 1 was more characteristic of those with a fatal outcome to COVID or to SARS-CoV-2 infection, whereas the microbes on this side are associated with protection with more mild disease, and even in those with severe disease, these are the people who recovered. And this is actually, this was really surprising, but nicely surprising, because actually the microbes associated with more severe disease are typical microbes that we don't want too much of, like Enterococcus or Streptococcus, whereas the microbes that are associated with protection are microbes that we know have immune regulatory effects. And in particular, I just want to point out Rheumococcus. So there's many different Rheumococcus strains in here, and Rheumococcus are very important for fermentation of dietary fibers. So this kind of fits with, there's more of these microbes that can ferment dietary fibers in those who do well. And when we put all of this data together, all the immune cell data, all the metabolite data, and all the microbiome data, we can generate these models whereby we can look at what are the interactions that really characterize someone at high risk of a poor outcome to SARS-CoV-2 infection, and those interactions that characterize someone who will be protected. And I'm just showing you here that interaction map for those with a poor outcome. And here are the cytokines like TSLP and IL-15 that I've already indicated to you, the microbes like Enterococcus, and also a lot of the metabolites from microbes like tryptophan metabolites, and also some other biogenic amines as well. So this shows that these interactions, and this is what we're researching right now, is that these interactions may be very important to promote the appropriate immune response to SARS-CoV-2 infection from an effector point of view early in the infection, but also control that exaggerated inflammation later in the infection. I'm just going to summarize that here for you. So on the one side, we can see there's a low risk microbiome configuration. This is associated with high levels of regulatory and effector molecules and low levels of pro-inflammatory molecules. This is associated with one specific type of immune configuration as well, that we think that means that when you see something like SARS-CoV-2 in the lungs, you clear the virus and you protect the host from hyperinflammatory responses. But on the other side, you have people with a high risk microbiome configuration. And this is associated with lower levels of regulatory molecules, but higher levels of pro-inflammatory molecules. You have a different type of immune response. And what this means is that you get delayed viral clearance and you don't prevent these hyperinflammatory responses when you get infected with something like SARS-CoV-2. We think that what's underpinning these high risk and low risk microbiome configurations could be dietary. We didn't look at any dietary questionnaires or FFQs in our data, but other people have. And I just want to highlight this one study, this really nice study that looked at people in the UK and in the US. And they did this diet quality score, which is based on a plant-based diet. So people got a higher score if they were eating more whole grains, fruits, and vegetables, and then lower scores for more refined processed foods. And what they could show is that those who had the highest plant-based score, or sorry, those with the lowest plant-based score had a higher risk of getting COVID, and also in other analysis they showed higher risk of getting more severe COVID. And I think what was really interesting, and we don't have an answer for exactly why this might be, there was an interaction with socioeconomic deprivation, so that in those who were in deprived socioeconomic situations or environments, that this dietary interaction was even more significant. So perhaps some of these, these dietary factors like fruits and veg and whole grains could be really important to drive a protective configuration of the microbiome. And possibly by generating things like short chain fatty acids that I showed you earlier are generated by fruits and veg and these non-digestible oligosaccharides and fibers that are fermented by the microbiome and can generate fatty acids. So what can we do about all of this? And this is something that this is where we are today, is trying to figure out, well, what can we do about this? One question I have, and I don't have the answer for this, is will dietary interventions on their own be sufficient? Because there is data where people have supplemented diet with fibers, for instance, and you don't see the expected metabolite or immune benefits. And that might be because some people have already lost the microbes that we required to ferment those fibers to generate these immune regulatory molecules. And I do think overall we will need to also consider not just dietary interventions, but also microbiome interventions. And this is just one example from a study we did a couple of years ago in influenza in mouse models. This is a H1N1 Puerto Rico 8 isolate that's mouse adapted. And you can see by day 9, all the placebo treated animals are dead. But in the bifidobacterium treated animals, you can see over half survive. And if you look at viral titers in the lung, it's very impressive difference between the two. And if we look at immune responses in the lung, I guess just to summarize this, is that those treated with the bifidobacterium had a very early, well-regulated immune response, whereas those that didn't have the bifidobacterium had this later, very damaging immune response that was associated with the fatal outcome in the animals. So I guess overall, what I would like you to take away from today is please do understand that many of the factors that shape our physiology and shape our immune response are actually non-human encoded. The microbiome and diet are really important parts of that. And please also don't think of the immune response to microbes simply as a form of host defense. There's an awful lot more going on there. And when we think about the interaction between diet and microbes, it's really that functional capacity and the metabolic outputs that I think may be the molecular alphabet of those interactions that underpin how the host immune response is influenced by the microbiome and by diet. And we really do need to better understand these communication networks, because we're only scratching the surface, really. We know about short chain fatty acids. I'm sure there are many, many other molecules that will also be important. And I think today we do have to be mindful of that, particularly due to the very necessary and important things like lockdowns, that we may have driven changes, maybe not just in the early life microbiome assembly, but maybe in all of us, because constantly you need to, or it's good for you to be exposed to new microbes. And we do need to understand that. And it's not to say that we wouldn't support lockdowns where they're necessary, but I think by understanding that, you can compensate for the effect of lockdowns on microbiome assembly and immune regulation, perhaps through dietary supports or probiotics or so on. And finally, I do think, and this is up for debate and for discussion, and I'd love to hear your feedback, is dietary interventions on their own may not be enough. We may also need to combine with microbe replacement, and how exactly we do that, that's what we need to figure out. So finally, I do want to just thank many. As you can imagine, there's been a huge number of people involved in the studies that I've been talking about, people from University College Cork, from the infectious disease people at the Cork University Hospital. We've also had a long-standing relationship and interactions and collaborations with many people in Switzerland, in the Kantonsspitals in St. Gallen and in Geneva and in Ticino, and in the Swiss Institute of Allergy and Asthma Research in Davos, and also to colleagues in the Karolinska and to Karina in Colorado and Carrie in Stanford. So I hope I haven't gone too much over time, and I'm very happy to answer, or to maybe not answer, but at least to discuss some of the questions that you might have. Thank you.
Wonderful. Thank you for this excellent update on the state of the science. As you mentioned, we can now shift into addressing some of the many questions from our audience. We've got some COVID-19 specific and some non-COVID-19 specific. So I'll start out with a couple of COVID-19 specific questions, if that's all right.
Sure.
So in those subjects with long COVID that are part of your study, what kinds of symptoms were they exhibiting, and were those expected based on the cytokines that remained elevated?
Yes, so we have kind of selected a group that is really characterized by those fatigue and brain fog type of symptoms. As you know, long COVID is really a syndrome with many subgroups. The subgroup, who have lung problems or long-standing lung problems, they're probably mostly due to the damage caused by the acute infection. So we didn't really want to focus too much on those. So we've really focused on those with the fatigue and the brain fog, the more unexplained symptoms that that the microbiome and the immune system might be more important in. The cytokines that we saw were elevated. I'd love to say that we predicted, that we were wise and we knew that we could have predicted they were elevated, but they were a surprise. It's a surprise that we appreciate, because it is pointing us towards there's still something going on in the epithelium, and there's still something going on with the immune system, that the immune system is still active in some way. And that's a very general statement, but there is still something that the immune system hasn't returned to baseline.
Very interesting. Thank you. The next question is, considering the changes that have been detected in tryptophan metabolism with COVID, do we know that these are clinically relevant changes?
So clinically relevant, so I suppose so we suspect they are, just by associations, but associations are just that. I think the next steps to really define if they're clinically relevant or not is to intervene in some way. Uh, and I think particularly in those with long COVID, perhaps we have more of an opportunity in the near term to do that. So for instance, you know, people are, there's been 2 studies looking at selective serotonin reuptake inhibitors during acute COVID. That's, and what they do is, you know, they prevent the, or I suppose they allow for lower levels of serotonin still to be active within the system, and they have shown to be protective. So that would fit with our data that these decreases in serotonin might be clinically relevant. So that's one way to think about it. Other people have asked me about, you know, should we directly supplement with tryptophan? And I can't recommend that today, because if the metabolic system is geared towards converting tryptophan into, you know, uh, kynurenins and quinolinic acid, maybe just giving more will give more of these neurotoxic effects. So I think we need to perhaps think a bit more about how to do that.
Certainly sounds like an area for further study.
Absolutely.
Wonderful. I'm going to switch gear and ask some more non-COVID specific questions. The first one is, if a pre-existing microbial community is important for realizing the benefit of fibers, is early exposure critical for receptivity to dietary fibers?
Just that last bit, sorry, you broke up. Is early exposure?
Critical for receptivity to dietary fibers.
So I think the early exposure is really important. Absolutely. And I think, you know, the microbes that like these substrates, you know, even if a later diet goes through periods of, you know, not being so good, you know, those microbes will still be there, and they will be available, even if they're at low numbers, that if you switch back to a high fiber diet later, you know, at least they'll be there to be able to deal with that, and they'll increase in number to deal with that. And I also think that there's an early life window for setting the immune tone. And so having a lot of these metabolites early in life really gives a head start, you know, with the regulatory part of the immune system. So the immune system is more regulated early in life, that I think lasts for a very, very long time. So yeah, I think it's really an incredibly important window that we need to hopefully promote more of these fibers and fruits and veg within the first year of life.
Wonderful. I agree. The next question is, there are some efforts to understand microbial metabolites or signatures that could be used as markers of health. Do you see this as a feasible endpoint, or is it going to be much more complex? For example, specific metabolite signatures for certain people?
So actually, it's funny, we had a meeting about this earlier today. So the answer is, I think, is somewhere in the middle. The research is really looking at the moment using many of these AI and, you know, learning models about trying to look in the entirety at all of these metabolites. And, you know, the predictive power of those models by predicting what people are eating seems to be quite accurate. So, you know, it is meaningful to look at all of these. Um, but I think we have to simplify it, you know, to bring it back into, so what are the most important ones? You know, what are the top 10 that really are the best metabolites to indicate, you know, immune health, or you know, metabolic health, which are closely linked, but it may not always be identical what's good or what's really the top 10 for one and the other. So I think the future, yes, we will see, I hope, more easy diagnostic type of tools that can be used for health screening, and that can help to say, you know, you've really low levels of butyrate, you really want to do something about that, for instance.
Wonderful. So this is sort of related, I think. You talked about the benefits of dietary diversity as a way to support immune function, and we're wondering if you could further characterize dietary diversity.
Yeah, and I know there are many different definitions for diversity. I guess at a very simplistic level, it seems to be that, particularly around plant-based foods, um, that having more different types of plant-based foods seems to be good for the microbiome, seems to support a more diverse microbiome. And often a lack of diversity seems to not be good for the immune system. So these are very general statements. What we need to do though is within, because by saying diversity, we're saying, yeah, just try everything and we hope we have the right things in there in enough amounts. Where we need to go is, what are the components in there that are really, again, important to top 10, you know, plant-based foods for a given microbiome that are going to be key. And we have research ongoing to try and identify those, and I know many others do too. So I think diversity will be better refined in the future. But right now, it's just simply you get what you can in and just hope for the best in a way. But it seems to be not a bad idea.
Wonderful. It sounds like we need to stay tuned for your future studies so we can find a clear answer. That's exciting. I'll switch back to some COVID questions if that's okay.
Sure.
So this one is interesting. The question is wondering, with the development of the vaccine against COVID, if you know if there's any interaction of vaccination with microbiome configurations that you've seen.
So the data for the COVID vaccines isn't there yet, but it is there, for instance, for influenza vaccine. And the answer is yes, there is a microbiome configuration that seems to be really important for more effective, for influenza anyway, um, vaccine antibody responses. And what people have shown actually is shown it another way. So it's, for instance, if you take an antibiotic a couple of days before getting an influenza vaccine, the vaccine isn't that good anymore at inducing a protective response. And that's because a lot of the gut microbes get upset. You get a big change in, bile acid metabolism. You have a lot more pro-inflammatory molecules and less regulatory molecules. That, I suppose, one of the things I always talk about in other talks is immune chaos. And when something is in a chaotic environment, you're not going to really be able to induce the very precise immune response you want to vaccination. So antibiotics a couple of days before getting a vaccine is not a good idea, and it seems to be mediated via effects on the microbiome. But as you can imagine, there are a lot of people who are right now looking at that from a vaccination, from a COVID-19 point of view. But I'm sure the principle will stay true, that there is a better microbiome configuration and a less protective microbiome configuration.
Very interesting. Thank you. I'm afraid we only have time for one more question, and that there's so many great questions here. I'll ask the last one, more of a general question. Are we getting exposure to microbes through raw fruits and vegetables that are influencing the microbiome development versus cooked fruits and vegetables?
Yeah, that's a really interesting point. And so the raw foods seems to be an important contributor to the microbiome pool. And as you probably know, there's a lot of people look at fermented foods and how they may be contributing. They also seem to be important. Um, so the answer is yes, maybe the ideal diet at the end is to have some element of raw food in there, as well as some element of fermented foods.
Wonderful, a variety. Well, that brings us to the end of our time today. A reminder that access to the CEU certificate will be provided by email after the close of the webinar. Thank you to everyone that participated and offered the great questions, and an even bigger thank you to Dr. O'Mahony for taking the time to share your insights with us. Please do register for our next webinar in the series on the IFANS website, scheduled for July 21st, and have a great afternoon.